insightful and amazing talks. Today on this 31st webinar, we have with us another alumnus, Deepak Kumar, from the State University of New York in the US. So before I formally introduce him, I would like to invite our mentor, Professor N Mishra sir, to kindly say a few words about Deepak sir.
Thank you, Anas. In fact, I am more eager to listen to Deepak Kumar. He was a master's student of our department, and I think it is which batch you were there, Deepak?
I was there, sir, 2007 to 2009.
2007 to 2009. So almost 11, 12 years has passed, but after that he is absorbed because there are very few people who are getting their absorbed regularly. Now I see that one of our stalwarts, Professor Singh, has joined, Professor Gajendra Singh. Good evening, sir. Good evening, Professor Singh. So I think let's listen to Anas. You can go with the formal. We are all eager to listen. Interesting talk by Deepak. He's doing an excellent job in you.
Thank you very much, sir, for those kind words. I would now like to take this opportunity to formally introduce Dr. Deepak Kumar. Dr. Deepak Kumar is an assistant professor in the Department of Chemical Engineering at the State University of New York, College of Environmental Science and Forestry in the United States. He is also an adjunct scientist at Carl R. Whey Institute for Genomic Biology at the University of Illinois. Dr. Kumar received his bachelor's degree in agricultural engineering from the Punjab Agricultural University at Ludhiana in India and has master's degree in post-harvest engineering from the Indian Institute of Technology, Kharagpur. He went to earn his doctorate in biological and ecological engineering from the Oregon State University. His research focuses on the development and application of sustainable bioprocess technologies for the production of high-value food ingredients, bioplastics, bioproducts, and biofuel using combined experimental and modeling approaches. With this, I invite Dr. Deepak Kumar sir for his presentation, and we're all looking forward to it. Thank you.
Thank you, Anas. So, can I share the screen now?
Yes, sir.
Yes, sir. Yes, please. Professor, by the time you share the screen, I would like to say good evening to Professor Singh. Good evening, sir.
I know, Dr. Gajendra Singh, good evening. It's great to see you.
You are muted, sir. We are really honored. Our laboratory lab team is honored to have you in this webinar. You are muted, sir. Sir, you are muted. I think there is some problem with you are still muted. Anyway. And good evening, sir. Good evening. Sir, we are really excited to have you in this webinar.
I know Deepak so well, I can't miss it. Okay. I don't know.
Thank you, sir. It's really, it's an honor.
It is honor for all of us and our lab mates. Yes, Deepak. Continue with the permission. Continue.
Go ahead, please.
Okay. So you can see my slides, right?
Yes, sir. We can see them.
Okay, perfect. So again, good evening, everyone. And first of all, I would like to thank Dr. Mishra for inviting me to present in this webinar series. I'm also thankful to all of you who are taking time to attend this presentation. And a special thanks, obviously, an honor to have him in the audience. So I'm going to talk today about sustainable bioprocess design using integrated techno-economic analysis. So what is techno-economic analysis when we use this term? So as the name indicate, it's made up of two words, technology and economics. So techno-economic analysis is a tool or a methodology to assess technical viability and economic feasibility of a process on commercial scale. So market commercial scale. So when we are talking about technical viability and economic feasibility, we're talking about now on the commercial scale production. So a product which we have designed or a process which we have designed, if we have to see how is it gonna be on the commercial scale before we actually do investment in that, that is where techno-economic analysis or TEA play a critical role. So let me show you an example. So recently my research group is working on bioplastics production from agro-food processing waste. So this is our recent publication in which we use acid whey, which is a waste stream coming from dairy industry, a big problem in the United States these days because of increase in Greek yogurt production, and there is no utilization of acid whey. So we developed a process through which we can utilize both lactose and lactic acid present in acid whey. We ferment it using recombinant E. coli, and you can see the cells. We get very high yield of polyhydroxybutyrate, which is a type of bioplastic. You can see the cells are filled with the granules. And then you take these cells, you extract these PHA, PHP out of it, and you can use that to make different kind of bioplastic products. So if you look at this, just this abstract, and the yields which I'm talking about, like 70% PHB content, 86% PHB content in the cells, this looks very interesting process. We are using waste, we are reducing the load on industry, and we are producing bioplastics, which will replace petroleum plastics, help in the environment, and in several other things. But the question is, what next? We have developed the process. We have showed that process is feasible on lab scale. You can make plastics. But where do we go next? Because when you will go to investor taking this technology, or you to go to the government or anywhere, there will be a lot of questions asked. Some questions like, is it viable on commercial scale also? Are there equipment available? Are there technology available? Is it scalable technology or not? What is the production cost of bioplastic? If you're producing kilogram of this plastic, what's the cost going to be? How it is going to be higher or lower compared to the existing plastic products? How much investment someone has to make on the front end to do this process? And many more such questions, for example, how much energy use, water use, labor uses, and those kind of questions. So these all questions we can answer using techno-economic analysis. And in today's presentation, very soon I will discuss a couple of case studies where we answered these questions, and those answers helped us further optimization of the process and help making that process more sustainable. So when I talk about technical viability, what do we mean by technical viability? Some examples are, what is the product yield? How much of that we can actually recover and get the final product? How much is energy use? How many waste streams we are generating? What is composition of those waste streams? And what should be the production scale up to which we can scale up this technology? Similarly, on the economic side, if we see, some of the matrices could be capital investment, annual operational cost, revenue, unit production cost, and obviously, finally, where everyone is interested is profitability, that how much is return on investment or internal rate of revenue. So this is techno-economic analysis. But when we talk about integrated techno-economic analysis, what does that mean? That means, like, when we are developing a process, it could be a bioprocess or any other kind of process, food process, and we are doing experimental studies. So what is our aim when we do design of experiments in the lab? Let's say you are making a antioxidant product, or you're making natural color product. So normally we try to maximize the experimental yields. We want to get that product, maximum productivity of that product. But if we can include TEA at the early stage of product development or process development, we can optimize our process, not just based on the experimental yield, but we can integrate energy requirement and economic considerations at a very early stage of process development. So it would be an iterative process. So we'll do some experiments, you input the data into models, do analysis, see if it is making sense or not, or what are the possibilities where you can make the improvement, and then you go back to your process, change the experiments, and then do more experiments. And in this iterative way, if we do this integrated analysis, the solution we will get will be sustainable, and it will be optimal based on multiple constraints, again, based on the yields, economics, and energy requirements. So other than just getting the overall economic matrices, this integrated techno-economic analysis, or TEA, it helps in a lot of other things also, which again, I go back to my title, sustainable bioprocess. How? Because it can help you in pathway selection. I mean, you all work on different research projects, and if you are targeting a particular product from a feedstock, normally there are several options. There are several pathway or conversion technologies to make the same product from the same feedstock. How do you choose those? So that is where this analysis can help you, because you can consider on the commercial scale which is going to use minimum energy, or whatever your requirements are. Based on that, we can do pathway selection. It can help you in understanding economic energy trade-off. So sometime you will, I mean, I'm sure many of you have encountered, there are processes, some are energy intensive, some require more economics, and you can balance those out. You can understand the trade-off and balance those out, depending again on the government policies and whatever your ultimate objective is. Identifying hotspots, that's very important thing, or the thing we get from TEA. So hotspot means we identify either the inputs or we identify the unit processes, which could be either energy intensive or require a lot of economics. So if we identify those, then the further research, we can focus on those hotspots to reduce the cost as well as reduce energy uses. Another thing it can help, excuse me, it can help us is deciding the scale of operation, because that's a very important parameter. We develop some technologies, but you need to understand, you need to answer the question that what is the scalability of that process? That would depend not only on the cost or the process, it will also depend on the material availability. So when you do techno-economic analysis, you include all those factors, and that helps you answering these questions and many more, many more such questions. So if we talk about basic steps in TEA, again, I'm just want to clarify that I'm not gonna talk about the process in detail, but as I mentioned earlier, I'm going to show you some case studies to show you the application of this tool and methodology in designing the process. But still, how we do it. So let us say you have a process technology in mind, or you have developed a process technology. So the first thing for TEA you will do is you will develop a process model. You will develop a computer model which include all details of your technology, every unit process, every single possible equipment, and you lay out those equipment in the way as it is in the technology. You set up the conditions, process conditions, and everything. So next thing is you do experimental studies where we need the data. More accurate data, if you can do the pilot scale studies, the data would be more valid for the scale up or the commercial process. But still, sometimes if we want to do analysis for very early scale studies, so we can use our experimental lab scale experimental study. So you feed that data, conversion efficiencies, compositions, recovery, and all that data into this model. Then you input cost data and assumption. So cost data is related to equipment cost. It is related to inputs cost, what is cost of chemicals, steam, electricity, everything, and the product you are producing, what is the selling price, and assumptions. Assumptions is a big part of techno-economic analysis, because you do not have all data available. So you'll have to take a lot of assumptions when you do that analysis. And that's where you will, people who will work in this area, you will find that assumptions are the kind of hurt which can decide how good your process is. You have to be very careful taking those assumptions. But after you input these things, then with the help of these computer models, you do mass and energy balance, and you do the simulations and analysis to figure out what is your process economics, energy use, and a lot of other things, which I will show you in a few minutes. So the where we do this process modeling, when I say we develop the process model, there are several softwares. The most common one are Aspen Plus, ChemCAD, and SuperPro Designer. These are the commercial softwares, but there are some open source platform also. One is BioSteam, which we developed a couple of years ago at University of Illinois, and there are several others where you can use, you need to do some coding, but you can do the same analysis without paying for these licenses. So the first case study I'm going to talk about is cellulosic biorefinery or cellulosic ethanol process. As you might know, that lignocellulosic biomass, such as agricultural residues, forest residues, dedicated energy crops, they are abundant in nature. They are available, they are of very low cost, and they contain carbohydrates which can be converted to sugars, and those sugars can be fermented to ethanol. So lignocellulosic biomass contain mainly three components: cellulose, hemicellulose, and lignin. And these three components makes a very complex structure, which is very analogous to concrete reinforcement structure we have in many countries. So you can see this cellulose, which is made up of only glucose. You can see it's the glucose linear chains, lot of hydrogen bonding. So they are in the form of macrofibrils. They just act like iron rods, you can imagine. And then there is hemicellulose, which is kind of a mesh you put around the iron rods in concrete reinforcement structures. And then is the most complex polymer lignin, which is kind of a concrete, you can imagine, or you can visualize it as a concrete you put at the end of the, in the concrete reinforcement structure. So these three components they form this recalcitrant structure. And the thing where we are interested for ethanol is these iron rods, because glucose, that's what we need. And of course, some C5 sugars also. So if you look at the process, overall process, again, you don't need to pay attention much on the details. But I'll just look at the overview. So you need to take biomass. You need to, ultimately, we are trying to get sugar molecule, glucose molecule, which we can ferment to ethanol. But to reach there, first thing you need to do is you need to break this structure. You need to remove this concrete or lignin and get your enzyme accessible to these iron rods or the cellulose chains. And that process is known as pretreatment process. So which has been focus of research over last couple of decades, and it is the most energy intensive process in the whole system. So what we do is we take biomass, we do the preparation, you do the pretreatment, which solubilize most of the hemicellulose, and which goes in the liquid fraction. Solid and lignin, they comes as the solid fraction, where you add enzymes which convert your cellulose into glucose. You do glucose fermentation. On this side, you do the pentose sugar fermentation, and then using distillation and molecular sieves, you recover ethanol. And the remaining fraction, which is lignin rich, you can either burn to produce steam and electricity, or nowadays there is a lot of research going on in converting this to biochemical. But coming to this particular case study, this was focused on this pretreatment process. There are a lot of categories of these pretreatment process. Could be you can use chemical at high temperature and pressure. You can use biological agents, physical forces, but the chemical one are more common and near to the commercial scale process. So you can see from these SEM images, like this is the untreated biomass, and this is after three different type of pretreatments: hot water pretreatment, dilute acid pretreatment, and alkali. And in all the cases, you can see the structural breakdown. So you have these fibers available on which now your enzymes can act, and you can produce glucose. So we developed 4 process model to compare 4 different technologies: dilute acid, dilute alkali, hot water pretreatment, steam explosion pretreatment. And we designed the plant assuming assuming the processing capacity of 250,000 metric ton per year. And you can see this is the which I was mentioning about the process model. So this is the process model of ethanol production. So you can see this has front-end operations where you receive the biomass, do cleaning and everything. This is the pretreatment section, conditioning section. SSCUF is simultaneous saccharification and co-fermentation. So basically we are fermenting glucose and C5 sugars together. Then you have product recovery section, and downstream you have wastewater treatment section also. Again, I'm not going to go into details, but just wanted to show you, like, through this modeling, we were able to, you can see, kind of mimic the biorefinery, actual biorefinery in the computer. So every process, every unit process, every equipment, and every details is embedded into these models. Similarly, for steam explosion, where all the sections are same except the pretreatment section. And then, as I was mentioning you, this was the we took the experimental data from another published paper in terms of conversions, compositions, and a lot of other things, and as well as some assumptions. So when you feed those, do the mass energy balance, we get these kind of results. So as I was mentioning you, economic matrices and energy matrices. So this, among the 4 technologies, this table shows you total investment, operational cost, ethanol revenue, and unit production cost. So I'll spend a minute here to show you what I was talking at the start of presentation. So now if you compare these 4 processes, let's start with the total investment. So among 4, if you see the total investment dilute acid process, this is high because you have to get the special equipment which are corrosion resistant, as well as you have to detoxify acid after the pretreatment section. So that add additional equipment. Okay, so in this way, steam explosion process is giving you minimum investment. Now, if we talk about operational costs are more in case of alkali process compared to dilute acid, and still minimum in case of steam explosion. So if you look at these 2 parameters, then you say, okay, steam explosion process is looking. But now you come to the ethanol production. If you see, in this case, your ethanol production is lower, where you are getting 15.8 million gallons of ethanol from the same biomass processing capacity. In case of dilute acid, you are getting more than, you're getting 14.1 million gallon, so about 1.5 million gallon lower compared to dilute acid process. And because of which, although your capital investment was high, but your production cost, ethanol production cost, is lower compared to this process. So now this analysis or these results have given you a clear picture. Like, okay, this is we're talking only in terms of economics, but you can, if you will go to this published paper, we did a lot of analysis on the energy also. Due to time constraint, I can't discuss everything, but idea is now it is up to investor. You give this data to them and they can decide which process we have to choose. Or if we were doing this analysis for research, then we know like which process is coming feasible and which process we need to do more research on so that we can decrease the cost in that process. For example, in steam explosion, now we know that you're getting lower ethanol. So we have a target, we have a research now, the new research direction that I need to increase this yield because I still have scope of increasing capital investment also. It's much less compared to dilute acid process. Even if I have to input some more chemicals or some other kind of energy, I can do that to boost up this yield so that my production cost will go down. So this was just the overall economics, where you can get also details like where your money is going. The operational costs are going into raw material, into facility dependent costs and other. And if you can see in most of the cases, the big fraction is towards the raw material. Within raw material also, you can get details like how much you're spending on tall fescue, which was our biomass, sulfuric acid, calcium hydroxide, diammonium phosphate, cellulase enzyme. So if you see, in all 4 cases, what we found was there were 2 main, the contributor. One was the biomass, and the second one was enzyme. So roughly 1/3 of raw material cost is going only in cellulase enzymes. So that gives us another research direction or another research question, that how can we lower this enzyme cost? Actually, this is what I did in my first year of PhD, and then the next 3-4 years, I did my PhD on reducing this cost, designing the synthetic cocktail of cellulase enzyme, which can have high efficiency and lower cost. Another thing is, as I was mentioning about the process selection. So if you, in this process, in the downstream side, after you recover ethanol from distillation column, the bottom portion, which is rich in unfermented components and lignin, what we were doing was we were splitting that between 2 sections. One part was after the filter press, the liquid fraction we were splitting between 2 sections. It could go to the multi-effect evaporators, where you recycle the water and get syrup, or you directly send it to the wastewater treatment section, where you produce methane and stuff, and then finally use that for heat and electricity. Now the question is, what should be this split? How much I should send to the evaporator? How much we should do for the anaerobic digestion or the wastewater treatment? So because we had the model ready, so we simulated model under different conditions, starting from 0 to 100% water going towards wastewater treatment plant. So, and then you can see the effect. If, as we are putting more water into the water treatment section, your capital cost is increasing, because your wastewater treatment facilities are capital intensive. So if you are ready to put more cost on the front end, it's going to give you a lot of benefit. You see the energy usage is decreasing, because multi-effect evaporator, they take a lot of energy. Those are energy intensive processes. So you can save energy. And once you save energy, you have extra energy available to produce electricity. Because what we are doing is we are producing in-house steam and electricity. Some of that we are using to meet the plant requirement, and the remaining we are converting to electricity and selling to grid. That was the assumption. So if your process energy decreases, you can have more electricity to sell to grid. So again, this is the trade-off, which the decision would be depending on what is our ultimate objective. Is it the investment? Is it the policies? Is it the research? But at least you can get this a clear picture using techno-economic analysis. So this was one study. I have another study to discuss, which is about co-production of ethanol and anthocyanins from colored corn in dry grind industry. So anthocyanin, I'm sure many of you know because you are in the food chemistry lab. Those are the pigments which impart red, purple, blue, these colors to the plant matter in the fruits and vegetables, in corn and a lot of flowers. So idea is, if you can extract these anthocyanins, these can be used as a replacement for synthetic dye in the food industry. Other than being a natural color pigment, these have several medicinal properties in terms of anti-diabetic, anti-carcinogenic, antioxidants. So it's a very valuable compound and can have a significant impact or the benefit if we can extract it and use in the food industry. So currently the main source is colored fruits and vegetable. However, this process is not very economically feasible currently because of large inputs you have to put in the material to extract those anthocyanins, because the anthocyanin concentrations are very small, as well as there is not enough material available. And the biggest problem is what you do with the remaining material after extraction. So let's say you take the blueberries, you take the blackberries. So if you try to extract anthocyanins out of it, what will you do with the remaining material? That is where this idea of anthocyanins from colored corn came into picture, and we did this study. So before going into that particular study, let's briefly talk about the ethanol production from corn. So this is the schematic of conventional dry grind ethanol process, by which in ethanol in US, most of the ethanol is produced. As you know, US is the biggest ethanol producer in the world, and we have more than 200 commercial plants using this technology. Idea is again same to lignocellulosic. You have corn, which is made up of starch mostly. So you have to break down that starch into glucose, and then you ferment that glucose to make ethanol. But in this case, you have the protein, oil, and other stuff also. So that down the remaining unfermented products, they are processed to produce animal feed. So briefly, again, you take the corn, you do grinding, you add water, and you form the mesh. The first step is liquefaction, which is done by adding alpha amylase enzyme. So idea of liquefaction is we're doing the starch gelatinization and then breaking down your starch into smaller molecules. And then next step is simultaneous saccharification and fermentation, where your glucoamylase convert those dextrins into glucose, and simultaneously that glucose is fermented to ethanol. Then you recover that ethanol, and as I mentioned, the remaining portion, which include fiber, oil, protein, unfermented sugars, that is processed to produce DDGS, distilled dry grains with soluble, used as animal feed. So if you look at the corn structure, which most of you know already, so there are four components: the pericarp layer, which is the outer layer of corn kernel, endosperm portion, germ, and tip cap. Most of the starch, that is in the endosperm portion, and this pericarp fraction, which is the fibrous component or fibrous layer, outer layer of the corn, that is about 8 to 10 fraction of corn kernel, mass based. So if you take kilogram of corn and if you fractionate that, you will get about 80 to 90 gram of pericarp, which is mostly fiber. So what we found, or what researchers found, was that in colored corn, especially in case of purple corn, if you see, most of the anthocyanin is concentrated in the pericarp layer. And in case of blue corn, it is in the aileron layer. So purple corn, which is rich in anthocyanin, it has already about 5 gram of anthocyanin, total anthocyanins per kilogram of corn. And a large portion of that is concentrated in pericarp of the corn kernel, which is easy to remove. So we came up with the process. This work is from University of Illinois with Dr. Vijay Singh. So they came up with the process where we do the front end fractionation. So idea, which is a well-developed commercial technology, low investment. So you separate out the pericarp. Now this pericarp is super rich in anthocyanin. So when we were talking about 5 gram of anthocyanin per kilogram of corn, if 70 to 80% is in pericarp layer, which is only 10% of your corn kernel, so you're going to concentrate anthocyanin 7 to 8 times in this pericarp. And if you remove only pericarp, your remaining portion is still intact. So your endosperm, your starchy portion, your protein and germ, which you needed for ethanol thing, that's still there. So idea was you separate pericarp, extract anthocyanins out of that using water or water with a small amount of sulfur dioxide in that. And the remaining corn you process through the normal dry grind process, grinding, slurring, liquefaction, SSF, distillation, drying. So which will not affect theoretically will not affect your ethanol yield. So again, a very cool process, but we wanted to check its commercial scale viability. So we developed the process simulation model. Again, all different sections: anthocyanin extraction, liquefaction, SSF, ethanol recovery, and downstream processing. We took the data from our lab scale studies, as well as used some assumptions. So one assumption was, because colored corn is currently not grown in United States, it's grown in the southern, it's mostly grown in Brazil and in the southern parts. So the assumption is that if you grow that in the United States, there will be some yield drag. So yield would be lower, so your price of this corn would be higher. So based on our discussion with farmers and crop scientists, we assume that the price would be approximately 25% higher compared to the yellow corn. And we did experiments with 2 extraction techniques, with pure water or using dilute sulfur dioxide containing 2000 ppm of SO2. And again, that all other data was taken from lab scale and pilot scale studies. So what we got, this is the process yield for dent corn, yellow dent corn, which is the corn used for commercial process, blue corn and purple corn. So if you see ethanol, it's a significantly reduction in ethanol. This reduction was not because of any process inefficiency. This was because of the composition of corn. So colored corn has relatively lower starch content compared to dent corn. So when starch is low, obviously your ethanol yields would be low. But you are getting a large amount of anthocyanin extract, which is a high value compound. And also if starch is lower, but the oil, fiber, and other components were higher, so their yield was high. Now, if you compare the economics of 3 processes, this is yellow dent corn, this is blue corn, and purple corn. If you see, the red bars are for capital investment and the blue bars are for operational cost. So both the capital investment as well as operational cost was higher compared to dent corn. So till now, we don't see any benefit because your ethanol is low, you have to put more investment. So, but when you do the complete analysis and calculate ethanol production cost, it's a significant drop. When you are producing ethanol at $1.30 per gallon in case of yellow dent corn, your cost reduces by more than 40% for purple corn, coming to only 75 cents. But case of blue corn, the cost increases because your yield is low. The reason for that is your yield is low, your investment is high, and you're not producing much anthocyanin also. Anthocyanin was more in case of purple corn. And that was the reason that your production cost was low, because if you see, although you're using raw material cost is more, utility cost is more, labor cost is more, but your co-product credit, the revenue you're getting from anthocyanin selling in case of purple corn is very high. And also the DDGS quality, because of high oil and protein, you have removed the fiber. So when you remove the fiber, there will be more protein in DDGS, which you can sell at higher value. So because of all those components, your co-product credits goes high and your overall production cost decreases. So, but from this, one thing you analyzed is that only purple corn is feasible. There is no meaningful idea of processing blue corn in dry grind process. So this answer or this conclusion you could not have gotten from lab scale studies, because on lab scale you were producing anthocyanin from both corns. But when you brought this TEA and did the commercial scale analysis, the results showed that only processing of purple corn is feasible, but not for blue corn. We answered some more questions. As I was mentioning about the extraction process, so the results I showed you were for SO2 extraction, because use of SO2 increases the anthocyanin solubility and its stability. So we were getting much higher anthocyanin yield. But if you do the simple water extraction, what we found was the ethanol production cost in both cases, both purple and blue corn, was higher compared to dent corn. So in that case, up till that stage, when we did that analysis, this process was not beneficial compared to dent corn if you use water extraction. So that led us to do the next study where we focused on the water extraction only, that how can we improve the feasibility of water extraction process. So we published the next study after optimizing the water-based extraction of anthocyanin and proving it with the techno-economic analysis. The last thing I want to show is, as I was saying, we also did this kind of analysis where initially we assumed that you can sell your corn at 25% higher price compared to dent corn. But we did the analysis by assuming like what will happen if we purchase the corn at 50% higher cost, 75% cost. So we found that even if farmer sell you colored corn 65% higher compared to dent corn, still your production cost is going to meet up to the dent corn. So basically, you can take a good amount of good hit on the yield drag, and still this process will be feasible. So now this kind of analysis gives motivation to crop scientists or the industries to invest more into this technology so that these colored corn varieties can be grown in United States also. So in conclusion, techno-economic analysis plays a critical role in research and process design. It can help in investigating commercial scale feasibility of new or modified processes, identifying challenges and hotspots, determining process conditions optimal based on technical and economic constraints, and identifying targets for future research. Last but not the least is it overall helps in making a bridge. So it bridges the gap between lab scale technology and commercialization, which is a big problem in research. We develop a lot of technologies on lab scale, but those are not commercialized. Even if they're commercialized, there is a time gap of several years. So this analysis can help in reducing that gap. And when you do that analysis at the front end, when you are developing your process, you are considering the commercialization aspects in the process design itself. So that gap will be reduced and the technology can be commercialized quickly. In fact, most of these days in the US, federal grant agencies, they ask techno-economic analysis as part of the proposal which you will use, so that you are not developing a technology only from the lab scale perspective, but considering the commercialization in mind. That's all I have for today. We have done a lot of other studies also, but which I can share or you can check at my website. And now I'll be happy to answer any question.
Yes, very nice presentation, Deepak. Excellent, in fact, I would say. So I think there must be some, I hope, many questions from the audience. So we can have, let's say, Anas, who is coordinating questions. There.
Yes, sir. We have one question in the chat box asked by Sindhura Makka. If you want, you can ask to sir directly, or I can read the question as well. So, sir, he's trying to ask that how is acid whey different from whey powder in the market?
Okay, it's a good question. So what happens is there are two type of whey produced in the dairy industry. One is acid whey and one is the sweet whey. So typical processing, dairy processing, which we do, that generates sweet whey, which is mostly lactose. So you can take that sweet whey, process it to produce lactose powder. But these days, especially in the United States, trend is going towards high protein yogurt, which is known as Greek yogurt. So when you produce Greek yogurt, you produce a lot of lactic acid also in the whey. So that's why we call it acid whey, because it's acidic, its pH is low, and it contains lactic acid also along with lactose. So presence of lactic acid limits the utilization of acid whey because it reduces its glass transition temperature. You cannot spray dry it will become sticky, and several other problems also. You cannot ferment it. A lot of microorganisms cannot ferment lactic acid. So it's a waste right now in most of the industries, and they have to dump it or pay the disposal cost for that one.
All right, thank you, sir.
That's a good question. And I think I partially answered that question in starting. So as I said, the accuracy level of these models or this analysis, that depend on the how comprehensive data you have and what scale data you have. So when we talk about there are various, I mean, again, as I said, this was just a short presentation. There are various level of analysis you can do depending on the accuracy level. Accuracy level can be from, in the techno-economic analysis, from ±5% up to ±40%. The models we are talking about is more for research perspective. So the production cost which I gave you, they are not, this is not actually the exact production cost you will get when you set up the process. But at least it gives you the idea in terms of comparison, the technology, in terms of the feasibility of the process, as well as we were saying, identifying the points where you can focus your research. Now, what you're talking about is about the product quality changes with the scale up and things. You can take those consideration when you simulate your model. So it's not it's a computer model is just a platform where you are setting up the process, but the conditions you have to set up. So all the process condition, product quality, what is going from one process to another, those all things we have to define based on the literature data, experimental data, pilot scale data. So computer modeling means just a platform to help in doing the mass energy balance. But the parameters, we set it up. And for example, I showed you DDGS quality. So it can help you in identifying in which stream, what fractions are coming, what is their viscosity, what is their composition, water content. So you can choose a lot of things, decide a lot of things, and analyze that data to get the quality analysis and stuff. And no, you do not need to do scale-up studies. That's what the point is. I mean, of course, it's good to do scale-up studies, but instead of doing scale-up study directly from lab scale, if you first put that data and use this TE in the early stage of development, your scale-up would be the variability in scale-up or the difference in the product quality you will get, or the optimization will be reduced. So it will be easy to scale up. And also, it's always not necessary to do scale up, because some things we know, like, okay, as you said, like, okay, when you're going from lab scale to high scale, you might have to do more studying. Maybe you need to maybe use a little bit different temperature. So you can adjust those parameters into the model simulation and get the predictions what will happen at the commercial scale or the large scale. I mean, obviously you have to, I mean, sorry, I just want to answer your last question. I want to answer one your question, and then I'll take next question. So the thing you mentioned about that we need to do some study, then only we can do for it. No, that's not true. Actually, we have published some studies based on the theoretical concepts also, which were actually very important. I'll just take a minute to show you one slide. Wait a second. It's important. It's a good question. Let me show you. So we did this study. You can see my slides, right? Okay.
Yes.
Yeah. So this was a technology. Basically, this is a recent technology, or I should say a recent approach in which what we're doing is we means like the group who was in or basically high yielding crop to produce lipids in those crops. So idea was you engineer those crops. So the carbon, you metabolize carbon instead of producing sucrose, that carbon was being metabolized to produce lipids, triacylglycerides. So this, when we did, idea was you can use that oil and sugar to produce jet fuel and ethanol. Because these crops, for example, sugarcane, that's the most productive crop in world. If you can put oil in that, theoretically, that comes to 20% oil, you can get several times, up to 15, 16 times high oil compared to soybean or jatropha. But idea was, will this process be even feasible if we have this crop? Is it going to be feasible? So we did this study where we developed a detailed model, assuming we have the crop in existence, and you process it. We know what would be the processing steps. You produce ethanol, jet fuel, and produce steam and electricity from the gas. We took assumptions. Based on the, again, discussion with the industry, based on our previous literature, that if you're converting sugars and lipids into these things, and we did analysis like, okay, what is the oil level you needed to make your process commercially feasible? Because right now you are in the development stage of the crop. So we started from 5%, 10%, 15%, and up to maximum 20%. And what we found finally was that you need to have at least 10% oil in that crop if you are gonna compete with the existing existing crops. For example, jatropha oil or jatropha fruit. So if you're producing jet fuel from that, if your production costs from this crop will be only match if you have 10% oil content. So many times this analysis can help you theoretically also in very early stage of development, when you haven't done experiments, but you have a concept in mind which you want to see how is it going to behave on the commercial scale.
Great. So, is there any, I think Sindhuja Makasi, okay, she has written something. Any other question?
Sir, I think Dr. Sindhuja only has this question. She has it. She says that in slide 17, the preferred mode would be hot water process, but is it the model which goes to production? If not, then does it depend on raw material, corn and enzymes, etc., procurement? Raw material procurement.
Sure. Yeah, I mean, that's again a good question. As I mentioned to you earlier, I have shown only the partial stuff of the study. You can refer to paper which gives more clear picture and detailed thing. So, but still to answer your question, as you said, the hot water process for that particular feedstock showed better economics in terms of capital investment, production cost, and everything. So yeah, we can conclude that yes, that out for that particular feedstock, this process is better. But it's not necessary that you cannot generalize it because the grass straw which we were using it will behave differently if you're using corn stover, switchgrass, forest residue or something. So, but you can use the same models, put the experimental data for other feedstocks and simulate to know which process will be better for that feedstock.
Thank you, sir, for answering the question.
Any other question? Thank you, sir.
Say something, sir. You are muted, sir. It was excellent. Very nice. You are doing very good work.
Nice presentation, Deepak, sir.
Thank you. And again, I mean, we have done this analysis for several food on this one. This is just one aspect. As I said, we integrate this thing into the development of processes. But my major research focus these days is not only on TEA. We are working mostly using agrofood processing waste, getting bioactive compounds, bioplastics, and other high-value bioproducts.
That's good. I see Dr. Bagh is also there. Dr. Bagh. Doctor.
Yes, sir.
So, would you like to say something?
Okay, sir. It's an excellent presentation.
Good. Very good.
Thank you, Park sir. As
okay.
I'm seeing you after, I think, after 12 years, right?
Yes, yes.
That time he was here in Kharagpur, no? When you were
Right.
Right. Yeah. So after a long time, but nice presentation. And
Thank you.
main thing is, I am also getting the interesting how the step by step, nice presentation. Further doing that things for scale upping and for which direction you have to choose. So really, it's a fantastic presentation. A lot of questions may rise. If the question will come, then it will be better for the students in which path you will go. And it's a better by one lecture. Students will not get the total things, but if you are interested, as Deepak told, lot of your in his website, you will get lot of information. And after that, if required any clarification, then Deepak can help you.
Yeah, absolutely. We collaborate with a lot of other researchers to help in their assessing their technology. So I'll be very happy to collaborate and help you in the things.
Excellent presentation, Deepak.
Thank you, sir.
Yes, sir.
Professor Mishra, I think
Sir.
this area need to expand in India.
Yes,
In
sir. Yes, sir. We are
agricultural
very nice, sir.
engineering, we have a very little activity in this area.
Yes, sir. Yes, sir.
And this is the area which is growing everywhere. Like I visited Vijay Singh's lab at University of Illinois. Okay, there's
Yes,
so much going on in
I
different
think
places.
we have now taken a few youngest in our department. They have some work in these areas. So perhaps in the coming days, definitely, sir. It is an emerging area, and we have to concentrate on this and do more work.
And you can
Your
connect.
team, Dr. Mishra, your team should submit a good project to NASF, National Agriculture Science Fund.
Yes, sir. Yes,
You
sir.
can
Definitely.
demand up to 5 to 10 crore rupees for the project.
Okay, okay, sir.
Look at the NASF ICR website.
Yes, yes, sir. I had one project in ASF. In fact, together with IIT Delhi and IRI, three of us were there. So we will definitely, sir, we will work out. Maybe we will talk to Deepak as well.
I'm a member of that committee now. Okay.
Sir.
I'll be very happy, Dr. Mishra, to collaborate. Yes.
Yeah, we can collaborate. So I will connect with you with sometime. We will discuss the things, and definitely, Sarkar आदेश है तो कुछ we will do that. Sure.
Yeah,
Because
no, absolutely,
this
sir.
area has a tremendous potential.
Yes, sir.
Yes,
Right,
sir.
sir.
And especially with the surplus production in agriculture sector in India, and energy is a big import for us. Okay.
Yes, yes, sir. Yes,
So there are a lot of options, a lot of pathways which
And
open
sir,
for us.
nowadays waste utilization, it will benefit in both the way. Sir.
Yeah, especially the rice straw.
Yes.
If we can look at that one, a massive problem and so much.
हाँ
many
पूरा
issues with it.
नॉर्थ ईस्ट में पराली का इतना जो प्रॉब्लम होता है सर दैट
Yeah.
कैन बी यस
And luckily we are growing rice everywhere.
Yes.
You can work on the technology
Yes.
anywhere and use it where possible. Okay. Very good.
Thank
Well,
you,
it's
Deepak. Very good.
minus 10 Celsius. With wind chill, it is minus 17. And I'm sure Deepak has a even cooler because he's north of.
Right, sir. Syracuse is like we get a lot of snow also along with
Sir,
lower temperature.
Deepak got so early to make this presentation, and we are equally honored, sir, that you also could make it. It must be in your place, sir.
Oh, yes, but now it's 8 o'clock, so it's okay.
Okay, 8 o'clock. But it's still 8 o'clock on Sunday. Is it
No, we're still Saturday.
Saturday? Is still Saturday.
And Sir Mishra sir, for me as assistant professor, we don't have any Saturday, Sunday or something. So they're all same.
Unless you get a tenure.
Yep.
Till you get a tenure, you don't have any differentiation. Yes.
Right.
Okay.
Any other question from audience? Anyone else? I mean, even if you don't have any question and comes to your mind later, you can email me. I can type my email address here.
Yeah,
I'll be very happy to.
That will be better, sir.
Yes. Zakir Khan, he is from
Pakistan.
So there is a lot of potential to work on these bio materials, bio waste.
Actually, sir, on your direction right now, we are working on Sebucthorn seed protein, sir. We are utilizing that waste, sir.
Yeah.
And that is my PhD work, sir.
Yes.
Good.
We have recently got one publication, and the next publication is in Food Research International. You are one of the co-authors, sir. There.
Yes, I know. Good. Now I am telling that yes, it is Professor Singh has really given us a good direction, and many IIT we are there, but other than IIT team seminar, people from CFTRI, other agricultural university, they are joining. So I think it should be one area, major area where all of us, research institutions, they should try to take up this work and take it forward. That's what. Good.
Yes, sir.
Sure, sir. As I said, I'll be very happy to help in any way.
Yeah, what I will do, I will,
yeah.
Deepak, I will connect you with one of my young scholar. And,
okay.
All right. Maybe also I will discuss with my faculty colleagues, those who have joined and new area. So maybe we will definitely work out some proposal, and then submit.
Sure.
So if there is no other question, so that Anas or who is?
Yes, sir. I'll invite. I would like to invite Ankit Rathod for a formal vote of thanks, and I once again thank everyone for a very nice discussion. So Ankit, just a second, I'll just present the slide.
Yes. Good evening to everyone. Firstly, I would like to thank Dr. Deepak Kumar sir for such a wonderful presentation. Like all the details were so intact and the flow of the presentation was so good that not even for a single second I was like distracted from any of the thing. All things are so connected that each thing was so easy to understand. Like it never felt that this technical economic analysis is a new thing for us. So it gives me an immense pleasure to propose vote of thanks for this 31st lecture of FCTL webinar series. I wholeheartedly thank our speaker, Dr. Deepak Kumar sir, for sharing his vast knowledge, starting from connecting us to what a techno-economic analysis is and all the steps involved in it, driving us through the case studies involving the ethanol production from Lignocellulosic Biomass, and how the anthocyanins extraction from colored corn can decrease the cost in the corns with incomparable patience. From all the attendees, I express my heartfelt gratitude to our mentor and coordinator, Professor N Mishra sir, for providing us this opportunity to gather knowledge in areas of expertise of our seniors via this webinar series. Finally, a big thanks to audience and all the seniors who found out time and participated enthusiastically in this webinar series to grace this session with their presence. We expect similar, in fact, a more response for our upcoming lectures. In the end, once again, thank you everyone who directly or indirectly participated and gave their valuable presence for the success of this webinar. Thank you.
Thank you, Ankit. Really appreciate it. And I as I said, it's always pleasure to work with in any way with Dr. Mishra, and Dr. Gajendra Singh was like, जैसे कहते हैं सोने पे सुहागा वाली बात. I mean, it was great to have him here. And thanks everyone who attended the presentation.
Thank you. I am seeing Deepak after 12 years. Thank you, Mr. Sir.
Thank you. Bye bye.
Yes.
Okay, sir.
Thank you,
sir. Thank you.
everybody.
Thank you, sir.
Thank you. Thank you.
Maybe after 15 days, we will have the another presentation. They will announce soon.
Yes, sir.
Okay, sir.
Bye bye. Bye bye.
Thank you.
Thank you.